My name is Susie Theroux. I'm a scientist at SCWRP, the Southern California Coastal Water Research Project. And so it's my pleasure to spend about the next 10 minutes giving what I would consider to be the California perspective on scaling and how we transition from research to implementation for eDNA tools. If you're less familiar with SCWRP, SCWRP is a public agency in Southern California that focuses on research to inform policy development and environmental management decision-making. We judge our success by how we transition tools from research to implementation, and so what I'll be spending some time today talking about is what I think are some of the real secrets to success of how we've been able to do that for eDNA-based tools. Really, I think the crux of it is something that Chris just highlighted, and it's relationships. And so what SCWRP really relies on is the relationships that we have with our regional management community, as well as the public agencies that are routinely going out and performing large-scale and long-term regional monitoring programs. Through these relationships with regional monitoring programs, we're able to co-design and collaborate and communicate on eDNA tools and technologies and how they might benefit their programs. We're able to test out new tools and optimize them and then standardize these methods. And we're also able to work with these partners in the environmental management community, in the regulatory or regulated communities, to identify what some of the key knowledge gaps are and then do the science research to help close them. For a little bit of history, this is something that Steve had touched on earlier. In 2002, SCWRP published a study that was doing an inventory of all of the ocean monitoring that was happening in the Southern California Bight. What this study found was that there were about 114 marine monitoring programs that were spending about $31 million annually to do ocean monitoring. About 10% of that monitoring was being done by federal and local government programs, and a whopping 70%, or 24 million of those dollars, were being spent by ocean dischargers who were collecting samples as part of their NPDS, or National Pollutant Discharge Elimination System, permit systems. And that was resulting in almost 160,000 samples that were getting collected annually, which included bacteria and effluent and water quality samples. So it was with that knowledge in mind that we really started to capitalize on this idea of leveraging regional monitoring that was already happening at scale. And it really reflected the boots on the ground, or in this case, the boats in the water, that were going out and collecting all of these samples and provided a wonderful opportunity for us to deploy and test new tools. It provided spatial and temporal scaling that otherwise we just don't have access to. As I mentioned before, it's a great opportunity to co-design studies to answer biomonitoring questions, and we do that alongside the agencies and the managers who are going to be eventually users of that data. We get extremely valuable feedback on how those tools are working and the ways that we can best optimize them to fit the purpose for those manager needs. And then we also identify ways to optimize, standardize protocols, and fill those knowledge gaps. So it was with that in mind that the Southern California Bight Regional Monitoring Program was started in 1994, and it's a long-term monitoring program that repeats every 5 years. We're actually in the midst of a Bight cycle right now, Bight 2023, that goes through 2024. It's an integrated, coordinated monitoring program that answers critical questions about environmental status and trends. And right now, we've been working on incorporating eDNA-based sample collection throughout all of the different, what we call Bight regional elements, of which right now there are 7, and they scale everything from harmful algal bloom monitoring to ocean acidification. This year, we have over 80 different agency partners that are involved in the sampling of these efforts, and there are thousands upon thousands of samples that are being collected. In 2023 alone, we had about 1,000 eDNA samples that are going to be collected alongside all this rich environmental metadata with which we can begin to establish some baseline information about biological condition and biodiversity in the Southern California Bight. Another great example of where SCWRP's been leveraging these regional monitoring programs to deploy and test new tools is through our perennial stream assessment. So back into the freshwater. Our perennial stream assessment programs are run through the Water Board's SWAMP program, or their Surface Water Ambient Monitoring Program. They go out routinely and collect a few hundred samples at stream sites all across the state every year. They're usually collecting bugs and algae and water quality data, which they're using for calculating biological index scores, which you can see here, and performing biological assessments for establishing biological condition at these sites and making their listing decisions as part of their Clean Water Act reporting requirements. For our purposes, we've been working closely with them to also collect DNA samples alongside their traditional microscopy samples. And with those, we've been able to compare how traditional microscopy and DNA samples are generating algae taxonomy data for developing novel DNA-based biological indices. We've also been using these samples to test and pilot the development of invasive and endangered species digital PCR assays. This in particular is a really big need for our management communities right now. They don't have the capacity, time, or money, or staff to be surveying for a lot of invasive endangered species, and so layering on a water eDNA component suddenly gives them access to biological information they didn't have an opportunity to collect before. Additionally, it's been a really great proving ground for us to test out different methods and what's optimal for when we have field crews that are hiking for days into the Sierras, and then we have to make sure that however they're sampling their eDNA sample, it's portable and it can be preserved over time before they can get it back to the lab and stick it into the freezer. Again, this program has really been successful because of the key partnerships and relationships that we have with the environmental managers and the field crews from the water boards that are going out every year and doing this and are now they've been trained up to be able to collect these eDNA samples as well. Another opportunity that we've leveraged through the Perennial Stream Assessment Program is in something called the Swamp eDNA Metabarcoding Monitoring and Analysis Project, or CMAP. And this was a collaboration actually with Jonah Ventures, where it started with a couple hundred Jonah Ventures kits that were sent to a number of different monitoring programs that included state and local and tribal communities that were going out to our stream sampling annual events, and they layered on this additional water eDNA sampling. What I think that this project was so successful in doing in highlighting the three core components in that they were all using a standardized sample approach. They were able to create a data dashboard that was making the data digestible so that regional managers who have very little environmental DNA background can be able to go to this dashboard and look up some of the data that they got from their samples, and that can be used to inform future follow-up studies. And it also leveraged the availability of these commercially available kits that made the sampling, lowered the barrier to implementation for sampling, and made it easy for these field crews to go out and collect this additional sample. One of the other real benefits of using these regional monitoring programs has to be able to invest in closing some of our DNA reference library gaps, which I think, right, I have to take a drink now because I said DNA reference library gaps. So as we know, there are some really huge incomplete DNA reference libraries that can be a real challenge when we're looking to do DNA metabarcoding, especially at scale, and it's limiting some of these eDNA tools from reaching their full potential. In order to get the most out of these DNA metabarcoding data sets, we need to prioritize closing those library gaps. And one of the ways that we've been most successful in doing that is, again, leveraging the power of these regional monitoring programs that are already going out to these sites. They're already collecting the benthic sediments, and now we can bring them back, sort the samples, and then generate those vouchered specimens, which we send to the Smithsonian where they get sequenced and that data gets deposited. This was in partnership with BOEM, Bureau of Ocean Energy Management, who's interested in doing DNA-based bioassessments for Pacific Outer Continental Shelf platform biomonitoring, and was just, again, a really great example of where we're leveraging these partnerships and the boots on the ground going out and collecting these samples. By layering in additional DNA-based samples, we can really begin to scale this methodology. I wanted to highlight also in California, we recently had $9 million that were appropriated for the Intertidal Biodiversity DNA Barcode Library project. This is really exciting. In theory, launches this year, a partnership between the California Ocean Protection Council and the California Institute for Biodiversity. We're working closely with museum partners at the LA Museum of Natural History, the Smithsonian, and the Scripps pelagic and benthic invertebrate collections for, again, identifying those priority gaps that we want to fill in DNA reference libraries and going out and vouchering those specimens and then sequencing them. And this is actually the third one of these barcode library gap-filling exercises that California has funded, I think, in the past 10 years. Previously, it was for insects and for fungi. And so I'm really hopeful that this serves as inspiration for other states or federal programs to prioritize filling these DNA reference library gaps, and I'd really love to see that be the next space race that we have for DNA reference library gap filling. I wanted to also mention that in addition to implementing these eDNA methods at scale, having these close partnerships with our management and public agency communities has also been a great way of closing knowledge gaps. And so this is really kind of the fun stuff where we're doing method intercalibration and optimization. You can see in this picture on the top left side of your screen, we have someone using a Smith Root citizen scientist sampler alongside one of the Jonah Venture sampling kits. And then they're also comparing that to vacuum filtration back in the lab. Because we're doing this alongside the managers, they are getting hands-on experience, they're getting training, and it's building trust in the method because they get to see how these tools operate in their hands, and then we have a growing user base for these tools. Additionally, we get to do more exploratory studies, such as looking at eDNA fate and transport, like a study that we did in this scenic Southern California river, where we were looking at propagation of environmental DNA downstream. And it's also been a really critical part to rely on our regional monitoring partners for method standardization. One of the best examples of that is in Southern California. We use the EPA standard protocol for enterococci beach water quality monitoring, which we've now transitioned to using digital PCR. And San Diego is the first county in the US that has implemented this eDNA-based approach for beach water quality monitoring. And they will close beaches using this method, so it's being used to inform management decision making. Scrip now has a study to implement this in City of Los Angeles as well. Okay, one of the things that we heard from Mike that's such a critical component of the eDNA national strategy, and I couldn't agree more, and that's on the collaboration and coordination among different researchers and agencies working on eDNA-based tools. In California in 2018, we founded the California Molecular Methods Work Group, which is founded by the California Water Quality Monitoring Council, which is a part of the California Water Board. And the California Molecular Methods Work Group was founded when we had a number of different agencies that were first interested in onboarding eDNA-based tools, and we realized that how they were doing eDNA sampling and analyses was siloed within these different agencies. So we wanted to have a forum where we could bring everyone together and identify what those sampling and data reporting best practices were. We could have trainings and informational webinars that talked about the tools and opportunities for using eDNA-based approaches. And also really just bring people together and have a clearinghouse where we could identify what potential research collaborations there were and really just build eDNA literacy among our regional monitoring communities, with tribal communities, with the research community. And it's open to everyone. And so we now have over 200 members that I think probably represent over 80 different institutions or agencies. And it's just been a really wonderful opportunity to hopefully build out the familiarity with eDNA-based tools, and it's lowered the barrier to adoption because folks have been hearing about eDNA for a couple of years, perhaps, before they decide to implement it into their monitoring programs. So I can't wait to see what the national equivalent of our California Molecular Methods Work Group is going to be, and I really look forward to how that's going to be benefiting biomonitoring across the US. So with that, I just wanted to wrap up and say something that we heard from Dr. Lubchenko earlier was that eDNA tools are ready to implement. We've had really great success rolling these tools out with our environmental management partners in order to test them and get feedback, build the training and capacity, and really identify where those managers can see the potential in the eDNA tools, and they already then have the experience to begin to implement them. As Adam mentioned, one thing that I really love to do is look for the bright spots, so the places where eDNA methods have been used to inform management decisions and use that as inspiration for how we can be transitioning these tools implementation as well. But it's critical to not wait until these tools are perfect, and so there's a lot of work that can be done in terms of optimizing these methods or figuring out fate and transport and decay mechanisms, but really we're missing an opportunity today if we're not going out and leveraging these opportunities that we have boots on the ground to collect these eDNA samples. They are irreplaceable, and the critical demands for biodiversity monitoring now necessitate that we don't wait to collect them. So I hope this was a demonstration for you all of how we can really be leveraging all the sampling that's already happening to sample eDNA at scale. And I thank you for the opportunity to share our California eDNA story, and I'm happy to take questions if we have time. Thanks.
Do have time for questions if anyone has any in the room. I guess, Susie, one of my questions is California is a huge state, and you have a lot of investment from the state that is outside of federal efforts. How do you see this playing out in other states besides California that don't have
Yeah.
as significant of coastline?
Good question. I think what I think is so successful from Squirp's perspective is that you don't have to be, covering large space and time. You don't have to be a monitoring program that's covering 1,500 square miles. You can be a monitoring program that's focused on one lake or pond, but they're still potentially going out and routinely collecting samples, and that's your opportunity. And so whoever the manager is there, or whoever's going out and doing the routine monitoring, if there's an opportunity to demonstrate the power and potential of eDNA, like, grab it. And so, yeah, don't miss those opportunities, no matter the scale, I would argue.
Amazing. Thank you.
Yeah.
Oh, we have a question up here in front, and we have a microphone to bring over. One moment. And if you can say your name.
Thank you. My name is Isabella Molitori, and I am a graduate student at the University of Maryland, Baltimore County. I formerly worked for a water agency doing salmon surveys in California, and I'm wondering if there's any work being done pairing salmon surveys with water collections.
Yes, definitely. And you'll actually be hearing about some great targeted species work through the California Department of Water Resources, I think in a session later this afternoon. But yes, come find me afterwards and we can connect some dots. Wonderful.
Thank you.
Okay. Any others? All right, with that, thank you, Susie.